So without further ado, I'd like to introduce our first speaker, Dr. George Demetri. Now, all of us in drug development know and highly respect the work of our next presenter. He's the director of the Ludwig Center at the Dana-Farber Cancer Center and Sarcoma Center, and the senior vice president for experimental therapeutics at the Dana-Farber Cancer Institute. He is also an associate professor at Harvard's Medical School. George's research focuses on the translation of scientific discoveries into targeted drugs for the management of sarcomas as a model for the solid tumor research and development. This work led to the development of the breakthrough drug Gleevec, which we heard some about this morning, for the treatment of gastrointestinal sarcoma known as GIST, and paved the way for follow-on medicines such as Sutent. Today, George will present Speeding the Drug Development Process, Learning from Team Science, and New Partnerships. Welcome, George.
Thank you, Thank you very much. Hi, everybody. The clock is ticking, and I'm going to get rid of those postprandial doldrums because Senator Frist actually set this up beautifully. I did not pay him to do that. But the bottom line on this is that we are going to talk about some of the excitement of the last decade in the next 15 minutes. It was 11 years ago, this Time magazine cover showed up talking about some of the excitement of being able to personalize therapies for cancer based on understanding what was driving the disease, hitting some of the mutated genes with pills, leaving the normal cells in the body alone. So the question was this just a lucky one-off? And what have we learned? And what led to this advance? And how have we been able to change cancer research in the last decade? And I think it's important to understand, just like Senator Frist said, that many things are about medians, and yet none of us are median. It's sort of like Lake Wobegon. We are all above average. We are all individuals. So this is the old view of cancer. Simplified, cancer used to be thought of as a monolith. Then in the 20th century, we recognized cancer is not one disease. It actually matters if it started in the breast or if it started in the lung or if it started in the prostate. Cancer was different things, but this was by no means granular enough. And now we know that cancer is truly thousands and thousands of different diseases. Scares the pharmaceutical company because the market keeps shrinking, but actually we've reassured the pharmaceutical companies and the patients that we can actually still develop drugs because maybe not every cancer is going to be green, but we can actually find green tumors, blue tumors, pink tumors, purple tumors by this metric and actually put them all together based on what's driving the cancer. So I wanted to give you a little bit of my personal background. I didn't come from a medical family. I'm the first in my family to go to medical school and the first to go to college, for that matter. And when I was growing up, I always had somebody in my family who had cancer. And I was watching the suffering that went through that disease and the suffering that went through many of the horrible toxic therapies of the late 1960s and early 1970s. And I wanted to become a doctor to many ways to contribute to the fight on cancer. And what I put here was the famous quote from Pasteur was that, you know, chance favors the prepared mind. And I was lucky enough to go to Harvard as an undergraduate and Stanford for medical school, and I had lots of role models that prepared me for being able to look at cancer in a different way. And I wanted to point this out because this is what's great about a place like Harvard. We have excellence and depth across the board. Because when I was an undergraduate in 1977, my role models were these basic science endocrinologists like Claude Villet and Carol Williams, who worked on reproductive endocrinology and what makes insects molt, the kind of hormones that makes insects molt. And I love the idea of hormones and receptors and the whole idea of endocrinology that if you got too much of a hormone, you want to block it. If you got too little, you want to replace it. So that kind of very simple but effective mechanism really influenced my thinking about how we might approach cancer someday. Then in medical school, the whole idea of infectious diseases and watching this horrible disease called AIDS develop when I was a Stanford Medical School student, where in the San Francisco Bay Area it was just starting. And Tony Fauci, when I did an observership as a senior medical student at the NIH, was also one of these role models who started us thinking about resistance and all the problems of hitting different diseases that evolve in front of your eyes, like HIV was doing. And then when I got to Boston studying hematology and the whole stem cell hypothesis, I'm going to do a drop kick here to Doug Melton's talk later on in the day to talk about how blood production at the top of the blood cascade is a primitive stem cell that then goes through many steps and turns into all the different kinds of blood cells in the body. In 1986, when I got to Dana-Farber, I loved this cover of the New Yorker. And it is, if we blow it up, it is the stem cell approach to frozen desserts. And I use this as a way of explaining how blood production works, but also how cancer works. These things called cancer stem cells are somewhat ephemeral and something of a third rail in the scientific community. But the truth is there is a stem cell that gives rise to other more differentiated types of cells in the body, cells that grow up to do a job. And if they get screwed up, like we have here, if you have a normal stem cell that turns into, let's say, bone or fat or muscle or connective tissues, that's fine. But then something goes wrong in many of these cells, and they turn into various kinds of cancers called sarcomas. Cancers that come from bone or fat or muscle or connective tissues are called sarcomas. And in the 1990s, we realized that we could start to understand what made the sarcomas different than the normal connective tissues in the body. And that gave us the hope that we would be able to target it. Here's the other thing that happened in 1996, and I'm glad that Senator Frist mentioned this because not enough people recognize that this is the other revolution in healthcare. Information technology. Patients started turning to another doctor, Dr. Google. And what's the first thing that anybody who gets a serious illness do? They or their son or their granddaughter types this in. Tell me about my form of fill in the blank about the disease. For me, it's tell me about my form of cancer. That's important because Dr. Google can help us in our academic world and can help accelerate this whole process. Now, part of the fun of being in an academic world is that we have the freedom to think when we're not being pushed around by some of the market forces and have to do more with less and all that. But it is so important to identify important signals, and being in a rich and robust scientific environment helps link things that you might not otherwise think. This happens to be a mouse that should be all dark brown or silver gray, and what you see is on its head and on its tummy, it's white. It's white because during development it didn't get enough signaling from an enzyme that signals the color signal. What's interesting about this is that look at what happens if a young human in development doesn't get enough of that same signal. It's eerie how much this is similar. And when we were thinking about the signal here, which is an enzyme known by the 3 letters KIT, stands for that because this thing, if it's mutated, causes a sarcoma in cats. And Peter Besmer at Memorial Sloan Kettering, his check on immortality was to name this thing KIT for kitten. That's why this switch got that name. We thought this was a very important signal. This is what happens if you don't have enough of it. One question is, what if you have too much of it? Back to that endocrinology idea. You could either have too much or too little. This is too little. This is what happens if you have too much. A group in Japan with the molecular biology done by one of the guys on the last line who was in our lab at Harvard, he went back to Osaka, Japan and did the molecular biology on this, where they basically identified the cause of a human cancer being too much activity in this switch. It's as if you turn on a light switch and pour epoxy in the light switch. It's now locked in the on position, and that causes this cancer called a gastrointestinal stromal tumor, or GIST, GIST for short. We actually had a whole National Cancer Institute conference in the early days of this saying, should we call it GIST or GIST? Okay, let's do a soft G. Oncology's hard enough. We'll do a GIST. So this was our very first patient treated with this disease, advanced GIST, treated here at Dana-Farber and Harvard Medical School. That big red thing is an enormous tumor stuffing this poor woman's pelvis. On the bottom, you see how much those big white things are the pelvic bones. There's not a lot of room in there for anything other than the tumor. This poor woman was in a great deal of pain. She went on this pill, and a month later, we did this picture. A month later, it completely shut off, and you can see on the bottom, even on the CAT scan, how much it receded. Within a few days, her pain went away. She was able to sleep through the night, and we knew the drug was working. But it's nice to have pictures like this, although it did irritate my radiology department, because the head of our radiology department is from Belgium. She called me up and said, Georges, what are you doing with these patients? I just spent 3 hours troubleshooting my machine, because I didn't tell her we were testing a breakthrough new drug. So it's obvious that it worked. It's not a machine malfunction. This was for real. And the bottom line on this is that this is what used to happen. When these kinds of survival curves go down, that means somebody's dying. This was the survival curve rapidly going down before these targeted therapies were available, before the year 2000 when we started our work. You'll see on average that's 0.5. That means half of the patients are dying by 6 months. Here's what's happening now. Now, half of the patients are living for 5 years, an order of magnitude improvement. And about 20% of our patients still from this very first clinical trial in the year 2000 still come back to see me at Dana-Farber 12 years later, still taking this pill. First patient was treated here in the year 2000, FDA approval for this drug 2 years later, 2002. This, in some ways, is the message I want to convey. If we use the power of the science and we can really take that signal, have faith in it, that that is truly what's causing and driving the cancer, and then if we're both lucky and smart, would you rather be lucky or smart? I'd rather be both. If you're lucky and smart, you can then hit that signal, shut the cancer off, and this is the kind of speed we can then help patients with. In the process of doing this, and mind you, this was a disease nobody had heard about. All the key opinion leaders were telling the company that was doing this work, Novartis, that it would take us 10 years to find 20 of these patients. It turned out nothing could be further from the truth. The diagnostics improved so much while we were doing this that it turned out in the US alone, there's more than 5,000 patients, new patients a year, who get this disease. The diagnoses were being missed. They were being called something else before the diagnosticians got new tools to make the diagnosis. We also realized that this disease, which is only about 5,000 or 7,000 patients a year in the US, is not also one disease. There are different flavors of this disease. Different types of the disease behave differently. You'll see that some of them, the one with the KIT mutant exon 11, one version does very well for the longest period of time. Those with the no KIT mutation, those who are driven by a different signal, some of them do very badly, very quickly, and then others, you'll see how that curve just flattens out. Some of them are doing better for a longer period of time. Patients get this. At some of the meetings of these patients, at the advocacy group meetings, they literally will identify themselves this way. Hi, I'm Jane, exon 17. Hi, Jane, I'm exon 11. It is unbelievable how much patients actually get this ahead of the doctors. And when we talk about this era of genomics, it's very clear to me that we can explain this in a way that normal mortals can understand it. We don't have to obfuscate it behind excessively complex data. It's actually quite clear if it's important. Back to Dr. Google. This was actually hugely important back in the year 2000, 2001, because we knew that the most common misdiagnosis for this disease called GIST was something with a polysyllabic nightmare called leiomyosarcoma. And we actually called the Google folks and said, we have a life-threatening situation on our hand. In the US, about half these patients think they have a different disease. Could you guys help us by maybe tweaking your algorithm to let them know that if they search this, tell me about leiomyosarcoma, they get some information about GIST and they can maybe find the life-threatening clinical trial fixer. So here's what used to happen. Back in 2001, somebody typed this in and this was what was starting to come up. You type in leiomyosarcoma, you start getting GIST information. It could be that their algorithm was just machine learning that really worked quickly, that the machines were starting to recognize the GIST leiomyosarcoma connection and did that, or it could be that Google actually improved their algorithms. We'll never know. They'll never tell. But the truth is, we used this to accelerate our clinical trials. The NCI called me up one day, and when we started moving this from a Novartis-sponsored clinical trial to a National Cancer Institute national clinical trial, the NCI called me up and said, we've never seen anything like this. 100 patients a month are going on your trial. We don't see that in lung cancer trials. We don't see that in breast cancer studies. Where are these patients coming from? It is absolutely remarkable, the power of a good drug for a life-threatening disease and the kind of information tools that we have available now. Well, here's the other thing. None of us expected that this would work as well as it did. It worked in 9 patients out of 10. 9 patients out of 10 would rise from, like Lazarus from their deathbed. But there is that 1 out of 10. This was the 11th of our first 13 patients here in Boston. This poor fellow came in July 5th. He wasn't feeling better by August. We doubled the dose of the drug, still didn't help. And by September, you can see that those lumps outlined in pink and on the right in the PET scan, everything's getting worse. He was resistant to the drug. We expected drug resistance, and we were poised for this. So our patients were very generous, letting us actually sample the tumors so that we could learn from this. It was too late for this gentleman. The clinical trial did not help him. He went back home, died a few months later. But we learned a lot. We learned about what makes the disease resistant. And here's one of the things. All of these have a mutation somewhere in this thing that looks like a periscope pointing out of the cell. This thing, if the cell membrane is that white line there, like at my belt line, and I am KIT, the outside is scanning the environment like that. The inside is the enzyme that's kicking the other things in the cell. So if I grab onto something, I'll kick something else. And the whole thing stimulates the cell to stay alive and to grow. So this is the issue. What happens is as the tumor cell evolves and as resistance kicks in, you start with that one resistance mutation, and then you get others. You get others that interfere with the ability of the drug to shut this thing down. So we put out an all-points bulletin to all of the possible collaborators in the academic and biopharmaceutical space. Here was the first drug, imatinib is Gleevec, that's the generic name. Here's the new one we were testing, Sunitinib. You can see a lot more red on there. And what this really is this new drug hits the target even better. That thing, that big red bubble around KIT is bigger. It's more powerful, and it hits more bubbles. So we said, let's test it. We had the only resistant GIST cell line in the world because of my colleague here, Jonathan Fletcher, who was growing then in the laboratory, put them on the cells in the dish, killed them, took it to a clinical trial. I wrote it over Christmas 2001. First patient went on in February 2002. This is that patient. Gleevec never worked for him. All that black stuff is bad, symptomatic cancer. 7 days after he started this pill, this is what happened. Everything shut down again. He felt better within 3 days. This was another home run. First patient treated 2002, did the worldwide clinical trials. That little biotech that came out with this was ultimately bought by Pfizer. And we worked with this worldwide trial, got the drug approved worldwide in 2006. And then we did the science to say, why was it working better than Gleevec when Gleevec failed? What you're looking at in blue up there is the drug itself fitting into the pocket of this switch. It fits into the pocket like a hand fits into a glove. It turns out that the resistant protein, that multi-dimensional thing there, actually puts out a little elbow in the resistant case, and Gleevec bumps into the elbow, but Sutent, because it's smaller, can slip under that, slip right into that pocket and shut the switch off. It's a beautiful story. You can explain it to a 5-year-old. So the science actually helps us not only to understand the drug, but now to develop next-generation drugs as well. So here's the latest partnership. In screening how these things work, there was a whole new class of drug that I looked at with that kind of technology of structural biology and say, something else should work too. This drug all the way on the right called Regorafenib, we tested it. The company Bayer said, you've done this before, try it again. Let us have the drug do an academic started clinical trial. We had the regulatory paperwork with the FDA. We tested it here in Boston, there in Oregon, here in Fox Chase in Philadelphia, and UCLA. All of us got together. We were responsible. At the end of the day, we did it very quickly. First patient 2010, done by December 2010. By August, we knew this stuff was giving us a signal of great activity. So what did Bayer do? They said, good, write the study. We'll support it to go global again. This is a complicated slide. We did the whole thing. Here are all the other centers now. Now we bring in lots of other places across the US, Europe, Asia, Australia. Finished it in 7 months, 2011, got all the data in January, and basically, here's what it showed. Huge differences. The lines don't go down as fast in the yellow group that got the new drug. That's the bottom line. People are able to control their disease longer, and basically, it's an effective drug. This is in at the FDA right now. This drug also just got approved by the FDA as the first pill that's an anti-angiogenesis drug, the whole Judah Folkman theory. First anti-angiogenic pill to treat colon cancer after chemotherapy stops working. So this is another wonderful example of translation of basic science into humans. So in bottom line, the mechanism's the message. We can now choose the right disease in the right patients. We can start to understand the variations in cancer, like you heard from our systems biology group this morning. And understanding resistance is critical to overcoming the barriers to cure. And in fact, it does take a team. It takes many teams, not just us on the clinical side, but the clinical side you never see, the pathologists and the molecular pathologists, as well as all the imaging people and the statistics, and of course the collaborators in industry. There's no better time for the academic-industrial interface to work than in this world of scientific translation. And ultimately, this is what we're going to have. We're going to have a Match.com, not just to find life partners. We're going to have patients. I'm a patient looking for my treatment. This is what people want, and this is what I believe our science can get us if we do our jobs well. So with that, I'd like to say thank you, and thank you for your attention. Now, do you want us to stay up here?
Stay up for a couple of questions. Thank you, and thank you. What an incredible story. I get chills every time I hear about what happens for patients at the hand of this amazing science.
Thanks.
You talked a lot about the industrial academic partnership. Can you talk a little bit about the role of government and maybe other partners in bringing such important advancements to bear?
Absolutely, especially now that we're all worried about government funding. I think government funding is critical to really provide that base glue that holds the whole system together. A lot of what we can do as academic industrial partnerships is really based on the fact that the academic world is solid, is going forward, that training continues, and is not subject to the whims of projects, let's say. I do worry a little bit that individual projects, especially when one's held to Wall Street expectations, can go up and down a little too fast and not necessarily be in jibe with the academic long-term view that we like to hold in an academic environment. So I think it's by doing this that it's easy, this kind of work, that it's easy to go back to Congress and say, here's how the fundamental research has turned out, how it really translates into improvements in human health, and the kind of improvements that you don't need a statistician to see. We're not talking a few weeks here and there, a little quality of life that you need a highly specialized instrument to show. This is really, you know, patients living 12 years later with a disease that would have killed them. So I think it's, again, putting together the pieces of philanthropy, government, academia, industry, all together to make a whole.
And you talked earlier about the relationship between kind of the monolith that we initially thought of as cancer and now this personalized approach, and maybe a little bit of resistance or fear, if you would, that big turns into a lot of smalls. Right. How has that affected the work that you do?
I think it's actually enhanced a lot of the collaborations in the cancer centers. We have to develop whole new systems. At a place like Dana-Farber, we have a breast cancer group that used to not talk to the lung cancer group, that used to not talk to the colon cancer group, and it's absurd because what we realize is when we have research meetings, everybody's doing similar things, and rather than duplicate efforts, we've learned that we can do it much faster if we try to develop a community of people who then feed out to the disease specialty areas. And I think it's a challenge for companies, a challenge for academics, to try to develop the kind of expertise that does cross those barriers, because everybody's career, I'm a sarcoma doc and a new drug development doc. Many doctors identify themselves as breast cancer docs and want to develop new drugs. So we need a community of people, back to the team approach. And I think our academic reward system really needs to start recognizing that. We were lucky with a lot of the work I just showed you because virtually everybody has gotten promoted, everybody's famous, everybody's happy. And I think that's what we need to do. We need to build our young generations and do that and share the credit across the board.
And you spoke of patient empowerment. It was really a theme that we heard earlier, how patients have really helped to support and drive. What things do you think are left for patients to do, and how might we encourage them and further engage them?
I think we can absolutely meet them as partners. It has to be that way because the patients have the disease, and the more we can get them on our side and us on their side, we have to align. We have been lucky in our field because most of the patient advocacy groups are extremely thoughtful. Many of them are doctors and PhDs themselves with the disease, who are able to speak articulately, not just say, we want access to a new drug, we don't care if it's not proven. That's just irresponsible. We know that many of the new drugs we put into people cause side effects. We have to prove robustly that they work. And our patient advocacy community has been extremely responsible to do that. And they feel that we look at them as equals. And that's the important thing. It's that mutual respect factor that goes into building this kind of a patient advocacy group that works constructively. They can help me write studies. They can help me explain the studies to the population. It's really useful, and they're on many of our advisory committees. For example, with our Specialized Program of Research Excellence Award, we have patient advocates on the advisory board.
So fully integrated and fully involved.
Absolutely.
Thank you so much.
Thank
Absolutely
you very much, Freda.
fabulous.
Thank you. Thank you.